Welcome to Mr. OD's second recording here on the topic of meiosis from Chapter 8. Meiosis is a two-stage process. The first part involves homologous pairs. Here we can see examples, and the chromosomes have the same length because they have the same content and sequence of information, that is, instructions on how to build or manage certain aspects of our nature, sometimes features of a certain part of the body, such as the eye or maybe the digestive system. The homologous pairs, one chromosome comes from each parent, and both of those chromosomes are present in all somatic, that is, usual regular cells, skin, muscle, bone, kidney, all are somatic cells. And even when the cells are just about to divide, as illustrated in this particular pair, this smaller pair here, that have now duplicated, now in the G2 phase of interphase, these are also homologous pairs. The next phase of meiosis, meiosis II, involves sister chromatids. The homologous pairs have separated in meiosis I and now exist as singletons. The cells are no longer in the diploid state. There's only one copy of each chromosome with two sister chromatids attached at the centromere. The presence of this butterfly type shape shows that the cell is in G2 and getting ready to divide. Here's one more time. The big story of meiosis with our mnemonic device up in the corner. The sperms, to remind us we're talking about gametes, specialized cells for sexual reproduction. Meiosis produces haploid cells, half the usual number of chromosomes, no longer the duplication. And then the key factor to remember that it is the separation of homologous pairs occurs first, and then the separation of sister chromatids. Here is a cartoon, maybe a stick figure version, a schematic of meiosis if you're into purely visual. In the S phase of interphase, the chromosomes are replicated, creating this sister chromatid appearance. Homologous pairs are separated in meiosis I, and sister chromatids separated in meiosis II. There I pose the question again to reinforce. That's right, the homologous pairs in meiosis I and the sister chromatids in meiosis II. The replication of the DNA occurs during interphase. So during the S phase of interphase, the chromosomes are replicated. They take on this kind of what I call the butterfly type appearance, and we see the presence of these two structures, which I describe as soda cans, two soda cans oriented perpendicularly to one another. That's right, these are the centrosomes, composed of two centrioles. A little quiz reminder of how things are proceeding. Does meiosis occur in the regular somatic cells? No, this is to create gametes for sexual reproduction. How many divisions does meiosis proceed through? That's right, two separate divisions. First, separation of the homologous pairs, and then separation of the sister chromatids. What is the state of ploidy of the daughter cells during meiosis? That's right, the cells are haploid now, only 23 chromosomes and lacking this coefficient in front of the number n. And then my point I keep repeating, what's separated in the first part of meiosis? You got it, homologous chromosomes. And in the second part, that's right, the sister chromatids. One more time, a little visual reminder. Only after the separation of the homologous pairs is the separation of the sister chromatids. Here, a little chance to notice the key differences between prophase 1 of meiosis and prophase of mitosis. The same condensation of the chromatin into chromosomes. The nuclear envelope disappears. The spindles are evident, but now the homologous pairs form up in their corresponding partners. That is, chromosome number 17 lines up with chromosome number 17, forming this particular shape called tetrads, a key element that biology students want to commit to memory. In this tetrad state, there will be a little exchange of genetic material in a process called crossing over that we will find out about shortly. Here's another illustration of the interphase cell moving from late G2 into prophase 1 with, here are the tetrads forming, telling us that this is meiosis occurring and not mitosis. Metaphase 1 occurs the same way. The chromosomes line up along the central axis there in what's called the metaphase plate, but the big difference is it is now homologous pairs lined up. If you see this kind of arrangement where chromosomes are lined up in a central axis in the illustrations, but they are pairs, it has to be meiosis that you're observing and not mitosis. Anaphase occurs the same way, except for now it's the homologous pairs that are separated. Notice the space in between the chromosomes, which are largely nicely clustered together in a linear format. However, this time it's movement of these sister chromatids that tells us this must be meiosis I and not mitosis. This is an illustration that will only occur in this phase of cellular division. Telophase 1 is pretty much like telophase during mitosis, except for now the cells are haploid. In this first division, the cells move into their gamete-like state, and perhaps the nuclear envelope does not reform. In humans or mammals, what would be the number of chromosomes in the haploid cells? That's right, it would be number 23. Good work. And here is the illustration showing the daughter cells from telophase 1. Notice the presence of the sister chromatids here and the absence of the homologous partner. And a summary of meiosis I, moving from producing 2 haploid daughter cells straight away in the process, and the homologous pairs have now disappeared, separated from one another. Another source of genetic variation. We'll learn about this separation in our next videos. Please join me for the next one. Thank you.